Hello and welcome to BIOS Frontier Science. We're thrilled to welcome Dan Nomura, University Professor of Chemical Biology and Molecular Therapeutics at UC Berkeley. To kick things off, Dan, could you share a brief intro with us? Thank you again for joining. It's amazing to have you.
Thanks for the invite. Yeah, I'm a professor of both chemistry and molecular and cell biology here at UC Berkeley. And my lab has been historically working on using covalent chemoproteomic approaches to tackle the undruggable proteome, which I think is one of the biggest challenges that we face in modern drug discovery. That still over 90% of the proteome is considered undruggable. And, even if we discover cool new disease targets, we can't therapeutically access them yet for most targets with really any kind of therapeutic modality that's available right now. And so we've been using our covalent chemoproteomic strategies to, really expand upon being able to drug a lot of these really challenging targets, but also to, expand upon new therapeutic modalities, whether it's expanding the scope of targeted protein degradation technologies that's become really, really popular in drug discovery, or going beyond degradation to develop new therapeutic modalities that are based in this kind of induced proximity approach that we take to manipulate protein or nucleic acid function, beyond just destroying these proteins. And so that's what we've been doing.
Fantastic, Dan. Thank you again for joining. I'm super excited to dive into your research. But before we go quickly in here, I love to ask a question just to start here. Before we get into specifics, just one question we like to ask our guests. Throughout your career, I mean, what has been your North Star, the common thread that ties your work together, if you will?
Yeah, so ever since I was a freshman at Berkeley, I think my first semester at Berkeley, I was still kind of lost as to what I wanted to do. My parents had wanted me to be pre-med and become a medical doctor. I think they maybe still want me to do that. But in my first semester, I quickly realized that what I really, really was interested in was developing kind of new disease therapies for debilitating diseases, whether it's cancer or neurodegenerative diseases. In my personal life, I've certainly been affected by, my mom getting, breast cancer now twice, and my dad suffering from a really debilitating frontal temporal dementia and neurodegeneration. What I realized very, very early on, even back as a freshman, was that most of these therapeutic targets were just not tractable directly through drug discovery approaches. They were, among this kind of undruggable class of proteins. And so I was really fascinated by this and, how one would overcome these challenges. And, nowadays you have so many different therapeutic modalities at your disposal, whether it's biologics, cell therapies, CRISPR, gene editing. Back then, I think small molecules still were really the forefront of drug discovery. And I remember, reading these papers from Ben Cravatt, who had at the time was at Scripps Research Institute and had just started this lab. This was in 1999, and he had first published these papers on activity-based protein profiling using these covalent probes, and I very early on realized the massive potential of these technologies that he had developed to be able to start really enabling drug discovery against this like dark genome. And so, while I stayed on in a lab that was focused more on, originally like environmental chemistry and pesticide toxicology, using, kind of similar covalent probes through undergraduate and grad school in John Casida's lab, I always kind of had this fascination, deep curiosity around enabling chemical biology technologies that would really overcome drug discovery. And eventually I went into Ben Cravatt's lab as a postdoc to really get into this whole chemical biology drug discovery space. And then went from there to start my lab back at Berkeley in 2011. So to me, it's been a very kind of linear progression from my original curiosity and interests and reading about kind of enabling chemical biology approaches back then to now, where these chemical biology approaches are now a driving force behind the cutting edge and drug discovery approaches now.
Thank you once again, Dan. I'm super excited to dive in more to your work. It's amazing North Star that you follow, and it carries so amazingly into your research. So I want to pass it over to my co-host here, Minju, to take over our first section and discuss more about your research.
Hi, Dan. Thank you so much for coming. We're really excited to hear more about your actual, the specifics of your research. So you're a world-renowned expert in chemical biology and also a pioneer in this field of chemoproteomics, where a major focus of your lab is discovering new therapeutics for proteins that are traditionally considered undruggable. To provide a little bit of context for our audience, can you provide an overview of what chemoproteomics is? And I know you briefly touched about this in your intro, but can you talk about why you were passionate about treating the undruggable? And in particular, what excited you about this chemoproteomics field?
Yeah, so there are a variety of flavors of chemical proteomics or chemoproteomics, but the flavor of chemoproteomics that we primarily practice is known as activity-based protein profiling. And this was really an approach that was pioneered by my former postdoc mentor, Ben Cravatt at Scripps Research, alongside also Matt Bojo at Stanford. And inherently, I would say the 1.0 version of this technology focused on active site directed covalently acting chemical probes, where these probes would be able to access the active states of large numbers of enzymes based on some shared catalytic, mechanism underlying an entire enzyme class. And initially, some of the enzyme classes that were really interrogated by this included, for example, the serine hydrolase family, the cysteine protease family, kinases. And what was really, really illuminating from those early studies was that you can, first of all, profile the activities of hundreds to thousands of enzymes, where many of these enzymes, even today, were among the uncharacterized classes of enzymes. So these were enzymes that had no known function, and we had no idea what they were, and what their physiological functions were. Yet we were still able to look at the activities of these enzymes and also develop inhibitors, selective inhibitors against these enzymes. And now, the technology has really evolved to be able to encompass really the entirety of the proteome, using more reactivity-based probes that can covalently modify, aberrantly reactive nucleophilic amino acids across the proteome, that not only take place in enzyme activity, but also really, a diversity of different types of pockets that might be accessible. And so these include things like allosteric pockets, protein-protein interaction sites, post-translational modification sites, and so on and so forth, that are usually considered more challenging or difficult to both find and also to pharmacologically access. And so in its modern era, chemoproteomics has really become a really powerful approach for tackling the undruggable proteome. So I guess per your other question, what is the undruggable proteome? So I would say that unlike druggable proteins like enzymes and receptors that have really well-defined binding pockets, an active site, or something that a small molecule can bind to clearly inhibit their function, undruggable proteins, which constitute the majority of the proteome, include things like protein-protein interactions or intrinsically disordered proteins, where it's really not clear where within the smooth interfaces of protein complexes or garbled, not well folded messes like intrinsically disordered proteins, which include things like transcription factors. It's really not clear where within these kinds of proteins you can identify, deep enough binding pockets for which you can develop a small molecule against to, functionally manipulate those proteins in a way that would give you, potential therapeutic benefit. And this has become a problem because, a lot of the low-hanging fruits in drug discovery among the classical druggable targets have been mined. There are so many well-defined disease-causing targets that we've known for decades are the major driver of these diseases that are in this, classically intractable, undruggable space. And not being able to access those has become an incredibly, big bottleneck in being able to develop game-changing medicines against many of these incurable or intractable diseases, and also these undruggable targets. And so one thing that chemoproteomics has been able to do is to be able to identify these pockets, these potential actionable, pharmacologically targetable pockets across practically almost the entire proteome now. And coupling that with covalent small molecule ligands, these approaches have now enabled not only discovering these binding pockets across proteins, but also the ability to be able to drug these proteins with these small molecule approaches.
Yeah, that's really fascinating. You briefly touched about this at the end of your previous answer, but can you talk about what chemoproteomics enabled covalent ligand discovery is and how the platform kind of works?
Yeah, so again, a lot of these approaches were really pioneered originally by Ben Cravatt and a lot of people coming out of his lab. But the approaches, these approaches are really founded on using promiscuous electrophilic probes that are able to broadly react across hyperreactive nucleophilic amino acid hotspots within proteins that could be involved in enzyme catalysis, could be involved in allosteric binding pockets or protein-protein interactions, any kind of binding pocket for which, there's a local protein microenvironment that's conferring lower pKa to these, amino acids like cysteines and lysines. And so these are promiscuous kind of, broadly reactive electrophilic probes that are able to fish these things out, bind to these targets. And then the way that we're detecting these probe modifying events is that we can, then append on some kind of enrichment handle like a biotin handle, and then subsequently pull down those probe labeled proteins. And then you can isolate these probe modified peptides, which you can then shoot onto the mass spectrometer. And coupled with really advanced proteomic approaches these days, you can really define exactly which amino acid or which protein target these things are binding to across, the entirety of the proteome. So, at its basic format, that's basically the readout where you're using these promiscuous electrophilic probes to find all these binding pockets across the proteome. And then what you can do with this is that you can have more tempered electrophilic covalent small molecule ligands that are going to be much more selective, and you can build out libraries of these. So our lab has built out libraries of thousands to tens of thousands of these covalently acting small molecules that have tempered reactivity and have a variety of substituents hanging off of them to make them more selective for specific targets over others. And you can deploy these in a competitive manner where you're competing the binding of these covalent ligands against the binding of a more promiscuous probe, either with pure proteins or directly in vivo and living systems, to be able to identify more selective ligands against specific targets in the proteome. And so, this could be deployed in a variety of different ways. So if you know exactly which target you want to drug, you can do target-based screens with covalent small molecule libraries to find binders against those targets. Those binders in themselves could be functional and inhibit or activate that protein, or they may be non-functional, but at least you have now a foothold into that protein and a small molecule ligand that binds to it. You could also deploy these in phenotypic screens where, for example, you're screening a covalent small molecule library in a biochemical or phenotypic screen. And once you identify hit compounds, you can use chemoproteomics for target discovery. So you can use these to, deconvolute the mechanism and identify precisely which amino acid and which protein target you're ligand bound to directly in a complex living system, whether it's a human or a mouse or living cells or lysates. So that's incredibly powerful. And another really powerful approach with chemoproteomics is the ability to assess the proteomoid selectivity of the molecules themselves. And so, oftentimes, I think one of the things that's held back this field of covalent drug discovery has been that these, electrophilic molecules, right, have the potential to be non-selective and can cause potential haptenization and autoimmunity and toxicity. And so a lot of pharma companies have historically kind of shied away from covalent drugs. But now with these modern chemoproteomic approaches, you can really understand the proteomoid selectivity of these molecules and then couple that with medicinal chemistry efforts to not only optimize potency of that molecule, but also its proteomoid selectivity so that the final molecule you get has very little off-target reactivity, and you can really mitigate and de-risk off-target toxicological liabilities. And so that's been an incredibly powerful aspect of this covalent chemoproteomic technology. So what this allows you to do, ultimately is to find binding pockets across really any protein target in the proteome, develop small molecule ligands that bind to it, and then be able to assess not only target engagement in complex biological systems, but also proteomic selectivity, so that you can then really, really optimize that molecule to be very, very selective for your target to hopefully, expand out safety profiles in addition to efficacy against that target.
Okay, great. Yeah. And kind of building off of that, your lab also has a pretty extensive machine learning platform in order to try to identify the selectivity, potency, and the toxicity of off-target with a lot of these compounds. So can you talk a little bit about that platform and how you use it in order to identify or select for some of these factors?
So I'll just say up front that so we don't have any machine learning capabilities in my lab. So Frontier Medicines is a company that I started based on this, kind of covalent chemoproteomic strategy. And in that company, we've deployed a computational team that's using machine learning approaches to be able to better mine our ligandability databases to be able to better predict ligandable sites that can be pharmacologically interrogated within these undruggable protein targets. And that machine learning team has further expanded their approaches to be able to, enable computational chemistry approaches to accelerate medicinal chemistry and all of that. But that's something that's not within my lab at UC Berkeley.
Okay, great. And I know we're going to touch on Frontier Medicines later on in this podcast. So as a follow-up question, it's really great to see that Frontier Medicines is addressing these needs. What do you view are some other problems that need to be solved in the field of chemoproteomics right now?
Yeah, so I think there's a lot of things that a lot of the people in the chemical biology field are working on to further expand upon these chemoproteomic technologies. So one limitation right now is that I would say the majority of covalent chemoproteomic strategies have been focused on targeting cysteines because there's a lot of different chemical matter that we know of that can react with cysteines, and many of these chemical warheads that can react with cysteines have been deployed into drugs that have made it into the clinic that are compatible with oral bioavailability and drug-like properties. And so these include, for example, a lot of the covalent kinase inhibitors that are now, sold against, for example, BTK or EGFR that are now approved. And also, more recently, the KRAS G12C inhibitors from Amgen and many other companies now, like sotorasib, that are kind of next generation covalent drugs that are more reactivity based, but still very, very selective for targeting a classically very undruggable protein target. And that was work really pioneered by both Keyvan Shokat and now Amgen and many, many other companies that have now entered the space, including my own company, Frontier Medicines, with a GTP state KRAS G12C inhibitor. And also Paxlovid, right? The SARS-CoV-2 drug is a cysteine targeting covalent, main protease inhibitor for SARS-CoV-2. And so cysteine has been really, really well mined. But other amino acids are still, more challenging, primarily because, they've been less explored. There's less chemical matter for probing them, and even less chemical matter that has been developed that is tractable for drug discovery approaches. Ideally, like an orally bioavailable drug that's metabolically stable that would have good PK and PD properties for deploying in the clinic. And so I think developing new warhead chemistry for other amino acids outside of cysteines is an area that is of a high interest for many companies out there in many academic labs. And you're seeing a lot of companies come out in this space that are tackling lysines, tyrosines, and other nucleophilic amino acids with new types of chemistry and in chemoproteomic approaches. Beyond that, covalent chemoproteomic approaches are very, very powerful in being able to access these targets, but they're restricted in nucleophilic amino acids that can be targeted with covalent chemistry. But there's obviously probably a large fraction of binding pockets that we're missing that don't have a conveniently placed cysteine, lysine, or some other nucleophilic amino acid. And so being able to access, the rest of this kind of, dark genome has also been challenging. And this is something where, again, Ben Cravatt, Chris Parker, and others have been developing really pioneering technologies around, photoaffinity based reversible, fully functionalized fragment ligands coupled with chemoproteomics to be able to both discover these, you know, um, binding pockets and develop reversible small molecule inhibitors against them. And a company that's come out of that effort is Belhara Therapeutics, that was founded by Ben Cravatt and Chris Parker and Stu Schreiber.
Great. Now I'm going to pass it on to Drew to talk about another aspect of your lab.
We really wanted to chat more a little bit deeper here, expand the scope of targeted protein degradation. I mean, really just using your chemoproteomic platform as an approach and as an application. So, I mean, another widely recognized focus of your work you are pioneering has been in the field of using chemoproteomics-enabled covalent ligand discovery to expand the scope of targeted protein degradation, in which this is an approach to degrade specific disease-causing proteins via small molecules. Just to start here, could you walk us through the process of identifying and validating target for protein degradation using a chemoproteomic platform? We'd just love to start in an overview here.
Yeah, so maybe I'll just take a step back and just describe the field of targeted protein degradation, which has really exploded in recent years as a major driving force for drug discovery. And where this all originated from, right, was pioneering discoveries by Craig Crews, who's a professor at Yale University, who came up with this concept of heterobifunctional degraders known as proteolysis targeting chimeras or PROTACs, where you can potentially also with Ray Deshay from at the time at Caltech and now at Amgen, where you could use develop E3 ubiquitin ligase recruiters and then link them via a linker to a protein targeting ligand to induce the proximity of an E3 ligase with a target protein, to tag that protein with ubiquitin and subsequently destroy it using the cell's trash can called the proteasome. And that was discovered a while back, but I think there was a huge revolution in 2015 where there were a lot of kind of simultaneous discoveries from both Craig Crews's lab as well as Jay Bradner's lab that was, at the time at Dana-Farber, and, he went on to be the president of Novartis Research, where, you could use this kind of PROTAC modality in not only cells, but even in vivo to be able to degrade whatever potential protein target of interest that was, causing disease. That then really exploded the field. Alongside a lot of the work that Craig Crews was doing, there was this other kind of storied history around this originally what started with a morning sickness drug called thalidomide, which ended up causing really, bad birth defects in kids of mothers that was taking thalidomide, where the mechanism of action really wasn't understood for a very long period of time. But ultimately, it was discovered that this thing was acting through binding to this E3 ubiquitin ligase called cereblon, and acting as what we now know to be a molecular glue degrader to degrade, at that time, proteins that were involved in limb outgrowth that then led to the birth defects, and ultimately Celgene and now BMS exploited that, developing thalidomide analogs like pomalidomide that ultimately and other drugs that ended up being Revlimid, that now engaged in the C3 ligase molecular glue degrader interaction to degrade cancer-causing proteins that led to really transformative disease therapies for multiple myeloma and other cancers. And now, this whole area has really exploded, in popularity. But one of the bottlenecks in the targeted protein degradation field has been that, we have over 600 E3 ligases in cells, but we only have E3 ligase recruiters primarily for two E3 ligases. One is cereblon and another one is VHL. And it's becoming clear that those two E3 ligase recruiters are likely going to be insufficient to be able to degrade any protein target of interest. And so we need more E3 ligase recruiters. But unfortunately, E3 ligases are also among this kind of, intractable class of targets. These are not necessarily all enzymes, even though they have that ACE name at the end. Many of these are actually, scaffolding proteins where they're bringing the rest of the ubiquitin proteasome complex in proximity to the substrates to be able to ubiquitinate them. And so really, this is more of a protein-protein interaction problem where you're trying to develop small molecules against these things. But again, chemoproteomics has really stepped up to, I think, tackle this. Our lab has been able to identify potential reactive cysteines that can be interrogated across over 98% of the over E3 ligases. And we've been able to use covalent chemoproteomic approaches to discover new E3 ligase recruiters that we can use in PROTAC and targeted protein degradation applications. This is something that's also been extended by, Ben Cravatt and Xiaoyu Zhang at Scripps and Northwestern, where they've been able to also discover covalent E3 ligase recruiters. And also, other labs and companies have been using DNA-encoded libraries to be able to expand upon new E3 ligase recruiters for targeted protein degradation. So, while these things were originally quite challenging to go after, in the last even 3 or 4 years, there's been transformative discoveries, I think, to really expand the scope of these things. Now, the other half of this has been in the discovery of molecular glue degraders, right? So PROTACs are a little bit more modular in design. So you take a small molecule that binds to your target and you link it to an E3 ligase recruiter. And there's a little bit of optimization involved there and linkers and all that stuff. But, the design conceptually is simple, right? But molecular glue degraders are monovalent molecules that, literally glue together an E3 ligase with the target protein to ubiquitinate and degrade them. And again, an example of that is thalidomide. But historically, almost all of these molecular glue degraders have been accidentally discovered or, discovered through phenotypic screens, and then, later were recognized as molecular glue degraders. And there haven't really been rational design strategies to be able to develop these molecular glue degraders. Ultimately, if you're able to do that, this would be potentially a much better strategy to be able to degrade proteins, because for the truly undruggable proteins, they may not even have a deep enough binding pocket for you to access to develop a ligand that binds to it to make a PROTAC molecule. The nice thing about molecular glue degraders is they're oftentimes able to access, the interface between protein interactions and cooperatively bring those together to glue them, even if you didn't have a deep enough binding pocket in either protein. And so it's a potential much more tractable approach to be able to access some of these really, really challenging targets. And so this is something where we've taken multiple shots on goal at this. So we've taken both phenotypic screening approaches with our covalent ligand libraries, coupled with chemoproteomic approaches to discover new molecular glue degraders. And on the flip side, we've also discovered covalent chemical handles that can be appended onto protein targeting ligands to convert these non-degradative, small molecules into molecular glue degraders of their targets. Hopefully trying to develop rational chemical design principles for discovering these molecular glue degraders in a more systematic manner. And so these are all different types of approaches that we've been taking using our covalent chemoproteomic approaches to expand the scope of targeted protein degradation.
Of course, it's an incredibly exciting overview of the research. We're so excited to follow in the Nomura lab as you continue to push down this vein. Just a follow-up question. I mean, it's been amazing to see how chemoproteomics has been able to follow on and add to that PROTAC wave that's been we've seen in the past few years. As a quick follow-up question, Dan, your lab, as you mentioned, we're currently using a few phenotypic approaches, covalent chemical handles. I mean, going forward, I would love to look forward here in the future for a moment. As the technology progresses, I mean, how do you imagine this design process changing, evolving as the technology and then future work progresses?
Yeah, so we definitely have a major emphasis in our lab on really enabling more and more technologies that will make this whole targeted protein degradation strategy more tractable, more modular, more systematic, so that we're able to apply this to potentially any target of interest, right? I think one of the challenges in the targeted protein degradation field has been that, while the promise of this technology is that you can potentially degrade any protein, especially the undruggable protein targets of interest, a lot of the molecules that have been developed so far have been against more tractable protein targets like kinases or steroid hormone receptors and stuff. I think what's very much front of mind is how do we apply these approaches towards the truly undruggable protein targets that are drivers of diseases. And so that's something that we're continuing to, push. I think going beyond that, though, not every protein benefits therapeutically from destroying it. A lot of proteins might benefit from potentially deubiquitinating and stabilizing it, or deacetylating, or phosphorylating, or dephosphorylating, or doing other things to it, right? And you know, you not every therapeutic target would even benefit from targeting the protein side. Maybe you might benefit from targeting, at the RNA or DNA level or epigenome level, right? And so I think one of the things that's been really top of mind for us and also a huge emphasis for our lab has been, how do we go after developing these new arsenal of therapeutic modalities analogous to targeted protein degradation, but going beyond that to be able to potentially, manipulate any potential protein post-translational modification or, protein, folding or even going into the nucleic acid space of being able to target RNA, DNA, epigenome with more fully small molecule-based approaches.
It's fantastic to see the future from your lens, Dan. I appreciate you sharing, and we're excited to follow on longer. You mentioned briefly on frontier medicines and some of your current applications that have spun out of the Nomura lab, but would love to pass it over to Minju for one of our last sections here, just to talk about in detail some of your amazing work you've been able to translate. Minju, feel free to take it away.
Yeah, so including Frontier Medicines, you founded a few other companies, but for today, we'd like to highlight Frontier Medicines because they focus on chemoproteomics, and as you touched upon, the machine learning platform to tackle these undruggable proteomes. So can you give us a little intro into the innovation behind Frontier Medicines, what it means to interrogate proteins in a living system, and specifically what the Frontier platform is trying to achieve?
Yeah, so one of the things that our lab had, developed over the years was that, we were running a lot of these covalent chemoproteomic experiments in our lab. And what we started doing was we started aggregating all of those data sets together, all of these, pro-modified sites. And when you do that and assemble them into a database, what you end up being able to do is really quantitatively map the reactivity of all these potential nucleophilic amino acid sites across the proteome. And collectively, we had identified over like 120,000 of these potential, ligandable sites across, the vast majority of the proteome. And that really core technology was underlying the foundation and formation of Frontier Medicines, coupled with the ability with covalent small molecule ligand discovery to be able to drug these sites. And so, very quickly we realized that we couldn't really, manually curate and mine this massive database that we had. And so we started applying, machine learning approaches to be able to predict ligandability. What is making a lot of these, cryptic, aberrantly reactive sites, reactive and ligandable, and what can we learn from these, and how do we apply those more systematically to enable drug discovery approaches? So, we brought together really, field leading experts in both, chemical biology, as well as, machine learning, with people like Dan Erlinson leading our chemical biology platforms and chemistry platforms, as well as folks like Johannes Ehrman, who's leading our machine learning approaches, coupled with an amazing CEO, Chris Varma, who was in venture for many years and helped start Warp Drive Bio and Blueprint Medicines. And now with a really rock star team of now over 100 employees at Frontier Medicines to really go after using covalent chemoproteomic approaches against high value, very, very well established undruggable drivers of cancer, right? So our focus at Frontier Medicines was really, really around applying these technologies to develop really game-changing drugs against already very, very well established drivers of particularly cancer that had just eluded drug discovery. And so, Frontier has been able to do amazing things with the covalent chemoproteomic approaches, and really, really proud of how they've industrialized, the kind of more academic platforms that we have in our lab.
Yeah, that's amazing. So kind of looking forward to the future of your company, what do you see as future directions for the Frontier platform, and where you see this technology evolving to tackle new challenges that are arising in drug discovery?
Yeah, so whether it's going after other nucleophilic amino acids beyond cysteines, or really, really making these approaches more high throughput to trying to develop ligands against every protein in the proteome, to really innovative chemical platforms to be able to innovate around covalent library design strategies. I think Frontier is really, really, I think, innovating across all of those fronts, and really, really proud of what they've been able to accomplish there. And then, just to also plug, my newest company, Vicinitas Therapeutics, it also underlying it is the same kind of chemoproteomic strategies, but now using those towards targeted protein stabilization platforms with these Deubiquitinase Targeting Chimeras that we've developed, or DUBTACs, where it's essentially the opposite of PROTACs, where we've now been able to develop covalent allosteric non-inhibitory deubiquitinase recruiters and link them onto protein targeting ligands to be able to, recruit a deubiquitinase to a ubiquitinated target protein to cleave off those ubiquitin chains and stop the degradation of key proteins for therapeutic benefit. And so this is, something that was based in covalent chemoproteomic strategies to develop these deubiquitinase recruiters, but has now been used to now deploy this new therapeutic modality for targeted protein stabilization. And so, that's kind of an extension of covalent chemoproteomics that's now housed in Vicinitas Therapeutics.
Yeah, great. So zooming out a little bit, more generally, either in chemoproteomics or other platforms, can you share kind of what your vision for the future of drug discovery is, and where you might see potential for either growth or innovation in this field right now?
Yeah, so what I'm really, really excited about these days is that I feel like with all these different innovations that have happened, both in chemical biology, but also, on chemistry side, the biology side, I feel like we now have all the different pieces required to be able to really blow out all of these different potential therapeutic strategies to be able to tackle the undruggable proteome. I think with advances in DNA encoded libraries and also covalent chemoproteomics, fragment-based drug design, I think this challenge of not being, of not being able to bind pockets and also develop small molecule binders against those targets, I feel like, we now have the raw ingredients to be able to do that now, to at least find binders against those targets. I think the challenge remains that simply finding binders against many of these targets is insufficient to modulate their function in a way that would make therapeutic sense. And so then what you need to complement that is the development of the corresponding therapeutic modality that allows you to exploit that ligand, in a way to, let's say, degrade a target or stabilize a target or do other things to the target, right? And so, I think now we're able to do that, and it's just a race to actually make all those modalities, develop these ligands, mix and mash them together, and make drug discovery much more modular and systematic to be able to now tackle any disease-causing protein target that we want to. And what's also really exciting alongside this, right, are all these other therapeutic modalities that have really arisen, whether it's immuno-oncology approaches, all the pioneering work from CRISPR-Cas9 and all the gene editing technologies that have come about from that, to cell therapies, right? I see all of these different approaches, right, as shots on goal to tackle the undruggable proteome, to be able to uniquely access all these previously completely inaccessible disease targets in a way that would benefit patients. And so I feel like over the next 5 to 10 years, this explosion in innovation is really, really going to transform drug discovery and allow us to make that currently 90% undruggable proteome to now being, 100% druggable.
Yeah, it's fascinating hearing your approach to kind of how you see the future going for this field. So to highlight some of your other impressive roles, not only have you been able to translate some of these amazing companies from your own work, but you are also an investment advisory partner at Andreessen Horowitz on the biohealth team, and also an investment advisor board at Droria Ventures. We would love to ask you a few questions about your intersection between academia and venture. So first, can you share how these advisory positions have shaped your mentality around forming new companies and entrepreneurship?
Yeah, so those roles are relatively recent for me, and I'm really, really enjoying working with Droya Ventures and Andreessen Horowitz. So Droya Ventures, I'm known for quite some time now. They invested in frontier medicines and also in Vicinitas Therapeutics, and were really early believers of a lot of these strategies. And it's been really wonderful to work with them, and ideate around potential new company ideas. Same thing with Andreessen Horowitz. They were one of our lead investors for Vicinitas Therapeutics, and the team that they have there, Jorge Conde, Vijay Panda, Benita Aguilar, Becky Furtifer, really, really creative people that are really, I feel like, pushing the edge of drug discovery, and really making really creative investment plays and really, really innovative sciences. And so being able to take part in that is incredibly fun. As an academic that's tried to do my best in trying to translate our work, being at the forefront of that in venture is something that I've been interested in for a long time, and now have the really privilege to be able to help these firms, with their investments.
Yeah. So speaking more broadly as an innovator, mentor, advisor at the intersection of academia and industry, we want to ask you a few questions about any advice you might have for young PIs seeking to translate their work from lab into an industry setting.
Yeah, so I think one thing is that it's very possible, right? I mean, there are many, many academics that have translated their discoveries into the clinic, and how you do that, there's many, many opportunities and different channels to do that through. So, my lab has major collaborations with various, pharma partners, and we can work with them directly to make these discoveries, and then they then deploy that into their pipelines potentially, or we can potentially spin it out into a startup company with whether it's NIH dollars or venture dollars or what have you. And so if there's, I don't think that this exists now, given the high degree of entrepreneurship that exists from academia. But if there's this concept that, oh, what you're working on at your academic lab is should be restricted to the academic setting and should be separate from translation, that's just a mental constraint that you're putting on yourself. And to me, back from when I was an undergrad freshman at UC Berkeley, my vision has always been that everything that I work on, I ideally would like it to be translatable and impact, the larger world, right? So really every project in my lab is actually designed to ultimately be hopefully translational. I don't think I have any really basic sciences projects in my lab. Every project in my lab is either hopefully meant to be deployable into pharma or be spun out into a new co. And so yeah, this is something that is basically a continuum for me. There's in my head at least, I don't really perceive a barrier between academia, industry, venture, startup creation. It's all just a single continuum.
Yeah. For other PIs that are interested on this entrepreneurial side, do you have any advice about creating a lab culture that is on this like more translational side of both science, industry, and venture?
Yeah, I guess, one thing that I think about a lot, both from uh, things that I work on in my lab to company creation, is especially as more of a tool builder, right? As a chemical biologist, where we're developing more like platform technologies for drug discovery. I think it's really important to uh, understand, is your technology going to really be deployable as a drug discovery platform that will yield actual drugs in a meaningful time frame, especially now in this current biotech climate, that would actually lead to a developmental candidate that would help patients, right? Oftentimes, and I've fallen into the same trap. You might create a platform technology like a hammer, right? A really cool hammer for which there may not be necessarily nails, right? So like a really, really souped up hammer that has no application. And so I think about that a lot in terms of when I'm creating a new platform technology, what will this actually be useful for in an actual disease setting, right? And am I creating a platform that is too over-engineered, that it's going to be too challenging to deploy as a drug discovery platform that would lead to an actual drug, or is this more something that's going to be niche to, more understanding basic sciences and be more utilized in an academic setting? So these are things that I constantly think of, even from the genesis of a project to once we actually substantiate the platform technology, how do we actually deploy this? Is this actually worth deploying into a pharma setting or drug NewCo setting, or is it something that is more suited just for academic settings? So yeah.
That's amazing, Dan. I actually just wanted to jump in here with a side question as well, as we're speaking on this intersection of academia, industry, with an emphasis of patient impact and translation. So aside from your industry appointments, I've noticed you're also an investigator with the Innovative Genomics Institute, an institute that is very geared towards translation and patient impact. I'd be really curious, among your perspective as an academic that's at this intersection, the impact or the broader scope of something like the IGI that has on your own research, as someone that is in this intersection and wants to create and generate more impact.
Yeah, so the IGI is an incredibly innovative research institute that was started by Jennifer Doudna, spun out of the discoveries that she made around CRISPR-Cas9, and she's brought together all these scientists across multiple different departments that are all focused on translation, either in human health or in agriculture. And honestly, for me personally, it's been transformative to be surrounded by people with the singular goal of translating these basic science discoveries at the bench into, the clinic or I guess in the field. I think there's definitely a space for academia to play in this arena, right? I think big pharma startups, they do their jobs really, really well in drug discovery and development. But I think a lot of these cutting edge platform technologies, whether it's gene editing or, the area that I work in chemical biology, are risky, right? And sometimes too risky for even, uh, immediately spitting out into a company or being deployed into pharma platforms. But academia thrives in these kinds of blue sky settings and just really, really nerding out over developing creative platform technologies. And to have that immediate infrastructure to be able to translate those discoveries, or at least get to higher levels of proof of concept, right? That gives you that added confidence that that this will actually be deployable into a more industrial setting. I think is really, really good. And it helps to bridge that valley of death in the middle of those like very early on basic science discoveries to something that can actually get capital and be deployable into the clinical settings. And so, yeah, I credit Jennifer for a lot of this. I think UC Berkeley has historically tended to be more basic science heavy, compared to translation. But I feel like the future of drug discovery is really around us technologists and developing these platform technologies and a lot of that pioneering effort is being done by chemists and biologists, right? Certainly CRISPR has been very much a basic science discovery that then very quickly spun into a translational endeavor. And Jennifer has really had the vision to bridge that gap between basic science discovery and translational sciences, and IGI is really the epitome of that representation. And so it's amazing to be here.
Yeah, it's amazing to hear from your perspective, the impact it's had from a cultural perspective, from an inspiration perspective, being around folks that are geared towards translation and have that focus. It sounds like it's been a key unmet need that is addressed and has been helpful to your lab. I mean, that's like really exciting to hear. And I'd love to interrogate that and ask a deeper question here. It seems like you had this mix between academia as fundamental research. You have industry that is constantly seeking to go for spinouts and have a translational side, but these research institutes are really good at that translation aspect. I mean, do you feel this was a key unmet need, something that was needed in academia? Do you feel this is something that will be more widely adopted or kind of the future of the space within academia? I'd love to know how you see this fitting into the broader ecosystem over time.
Yeah, I mean, that's a good question. And it's something that I've thought about a lot. I mean, there are a lot of these kinds of research institutes that have this kind of translational slant, right? And the question is, where does the focus of those translational institutes sit in relation to big pharma and biotech and also the venture community and everything else, right? And I mean, pharma does drug discovery and development really, really, really well. And academia shouldn't like just copy that because pharma has way more money to do that, and they do that incredibly well, right? And so where does academia sit in this, and where can they uniquely, I guess, contribute, right, to this? And I think it really comes down to the fact that in academia we have this what I've gained a renewed appreciation for is intellectual freedom and academic freedom, right? To explore these crazy blue sky ideas that may be too risky for biotech and pharma and venture capital, but we have the academic freedom to just try it out, right? And go into the lab and just tell our people to work on these crazy ideas, and a lot of times these crazy ideas work, right? And we can get the proof of concept and then get enough confidence that then pharma and VC and biotech get interested. And so I think that's the sweet spot, right, for a lot of these translational research institutes to be in that space that's yet unmet by institutional investors or pharma or biotech in this more challenging space, whether it's like tackling undruggable targets or developing new therapeutic technologies, rather than trying to recapitulate and go after similar targets that all the pharma companies are going after and doing that in an academic setting. I don't think that's that efficient because pharma's gonna put tons of money and tons of people into that area, and ultimately it's going to be hard to compete in that kind of space. So I'm very much a believer, and it's one of the reasons I've stayed in academia, of being able to operate in that blue sky space, be much more nimble, right, and be able to pivot as needed more quickly. And if something really, really takes off, then we can always spin that into companies or deploy that into pharma pipelines or what have you.
Wildly fascinating. I really appreciate the view from your perspective, Dan, on the evolution of the industry and tying these threads together for a focus on translation and how we can do that faster and more efficient. So this time has flown by. Thank you so much, Dan. Just one quick rapid fire question to cap this episode off. I mean, really just to close things here, any closing thoughts or shameless plugs that you'd like to share? I mean, how can our audience learn more about your work?
Yeah, I mean, first of all, my lab is always hiring. We're always looking for innovative chemical biologists to join our lab, and also Frontier Medicines in South San Francisco and Boston are also hiring, and we're always looking for talented people. My newer company, Vicinitas Therapeutics, is in South San Francisco and rapidly expanding to try to realize the potential DUBTACs and targeted protein stabilization into drugs. So for those of you who are interested in being in this kind of cutting edge translational chemical biology space, join my lab, join Frontier, join Vicinitas, or any new companies that I'll be developing.
Very exciting stuff, Dan. Love the shout outs to Vicinitas and Frontier and your own lab. Hope our audience enjoyed our time as much as we did. I mean, just thank you again, Dan. It was a fantastic episode.